In this lecture, you're going to learn about various types of synapses or junctions. A synapse is a junction between two neurons or between a neuron and an effector cell like a muscle or gland cell. In the example below, you can see a synapse between two neurons. There is a presynaptic neuron and then the junction known as the synapse and a postsynaptic neuron. It is important to be able to get the action potential from the presynaptic neuron to the postsynaptic neuron. In this case, the way that's accomplished is by a chemical being released in the synapse. This chemical is known as a neurotransmitter. There are various types of neuron-to-neuron synapses. The first is called the axodendritic. It's the most common, and it's a synapse between the axon of one neuron and the dendrite of another. An axosomatic synapse is also common. It's a synapse between the axon of one neuron and the cell body of another. Soma means body. Axoaxonic is less common, and it's a synapse between two axons. A dendrodendritic is less common, and it's a synapse between two dendrites. A dendrosomatic is also less common, and it's a synapse between dendrites and cell bodies. Here are illustrations of some of those types of synapses. Going from left to right, the axodendritic, remember, is between the axon and the dendrite. The axosomatic is between the axon and the soma or cell body. The axoaxonic is between two axons, and the dendrodendritic is between two dendrites. This represents a dendrosomatic synapse, that is, a synapse between the dendrites of one neuron and the soma or cell body of another neuron. I've mentioned these terms already, but I want to be sure that you have a full understanding of what they mean. And that is presynaptic versus postsynaptic. A presynaptic neuron is the neuron that transmits the stimulus toward the synapse. And if you look, the presynaptic neuron is the one on the left. It's conducting the action potential toward the next neuron. The postsynaptic neuron is the neuron that transmits the stimulus away from the synapse. Most neurons can function as both. That means they receive stimulus and they pass along that stimulus. We've talked about neuron-to-neuron synapses, but there are other types of synapses. There are neuromuscular junctions, which are synapses between neurons and muscle cells, and neuroglandular junctions, which are synapses between neurons and gland cells. These junctions can be either electrical or chemical. Let's look first at electrical synapses. They are less common than chemical synapses, and the transmission across the synapse is rapid. They can be either uni- or bidirectional and are abundant in embryonic nervous tissue. They're rare in adults because usually electrical synapses are replaced by chemical synapses. Places you find electrical synapses are the area of the brain responsible for stereotyped movements and in cardiac muscle. Chemical synapses are much more common than electrical synapses. You can see a diagram of a chemical synapse on the right. Transmission across synapses is slow, it is unidirectional, and it's the most common type of synapse in adults. This is a close-up drawing of a chemical synapse. I'd like you to be able to identify the parts that are indicated here. The answers are on the following slide. The green square is the ion channel. That's the place through which the chemical has to pass. The red oval is the receptor. A is the axon, B is the synaptic knob, C is the mitochondrion, which is responsible for producing ATP, D are the synaptic vesicles, E the synaptic cleft, F the postsynaptic membrane, and the green circle represents the neurotransmitter. Here are the steps in transferring an electrical impulse across a chemical synapse. Step 1. The action potential moves along the axon of the presynaptic neuron to the axonal terminal. Step 2. The action potential reaches the terminal and triggers the exocytosis, or the movement outward, of the chemical neurotransmitter in the synaptic vesicles. Step 3. The neurotransmitter molecules are released into the synapse and diffuse across the cleft to the postsynaptic membrane. Step 4. Neurotransmitter molecules bind to receptors on the postsynaptic membrane. Step 5. After the neurotransmitter molecules bind to the receptors on the postsynaptic membrane, this opens up ion channels in the membrane of the postsynaptic neuron. Step 6. Ions move through the channels and the postsynaptic neuron is either excited or inhibited. Step 7. The neurotransmitter must be removed or destroyed, and the reason is, as long as it's bound to the postsynaptic receptor, it's going to continue to have an effect. So how do we remove the neurotransmitter? Well, it depends on which neurotransmitter it is. The first one is acetylcholine. It's degraded by enzymes in the synapse. The second is norepinephrine, and it's taken back up into the presynaptic terminal. And the last possibility is the neurotransmitter can just diffuse away from the synapse. The synaptic delay is the time that it takes for the electrical impulse or the action potential to move from the presynaptic to the postsynaptic neuron. That is, the time it takes to cross the synapse. During this time, the neurotransmitter is released, it diffuses across the membrane, and it binds to the receptors. It is the rate-limiting step in the whole process, or the slowest step in the process, and it lasts for about 0.3 to 5 milliseconds.